Any questions? If anyone wants to post anything in the chat, or you can unmute and ask it yourself. I guess I'll start off with the question. Do you think that in areas that are kind of seasonally hypoxic or anoxic, that you have microaerophiles or even anaerobes that are like kind of hanging around at like a low level of abundance that then can kind of get enriched relatively quickly? And if so, how kind of quickly in response to kind of rapidly changing conditions that are anoxic or sulfitic?
Yeah, that's a great question. So I think that you're probably thinking back to the diversity, right, where you have a change in the evenness, right? So what I think you're getting at is it likely that these that there are microbes that are just background members of the community that you don't observe as being abundant, or if you don't sequence deep enough, you don't even see them, perhaps, right? I think this gets to everything is everywhere, comma, but the environment selects, right, from Bass Becking. And so I think that that probably is the case, that these microbes are there. They're just maybe not visible using the tools that we use to look at the microbial community. And so in terms of how rapidly they can respond, that's a good question. There have been a lot of studies in the Gulf of Mexico where they went to the environment before hypoxia had set in. And so, you're talking about maybe just a couple months before hypoxia sets in, and they see very low to no AOA archaea in that same exact environment where you don't have where you have plenty of oxygen. It's an oxic water column. And then as that stratification sets in and you have that bottom water, that Gulf of Mexico bottom water pinned to the bottom, you see a fairly, within a fairly short period of time, you see that bloom of that ammonia oxidizing archaea. So I can't directly answer the time scale very well, but I absolutely think that what I think you're suggesting, that, it's likely that these microbes that can carry out these their metabolism using low oxygen or can switch to a different electron acceptor are probably there. We just don't see them, or they're just very low in a very low abundances.
Cool, thank you. Kurt, you had your hand up.
Yeah, thank you, Olivia. Also, I enjoyed it very much. Two questions. What you talk about environmental drivers, you mean the macro environmental drivers, right? However, what you mean with communities are probably communities embedded in particulate sedimenting material, which might have a microenvironment that could be quite different from the macro environment. They could even be completely anoxic in there.
So in the particles, you're right. You do have this completely, this complete different environment than the water column surrounding that particle. But actually, Lauren presented that when we sample, we pre-filter out the particles. So we use a 2.7 micron pre-filter, and then we collect our microbial samples on a 0.22 micron Sterivex filter. And so we're not going to remove all the particles at that pre-filter size, but we are trying to look at plankton. So we're not looking at what's in the particles as much as we're looking at what's in the water column. So these are free living planktonic bacteria and archaea that we're focusing on here.
Mm-hmm. So you might lose an awful lot of bacterial biomass.
That is correct. So the particles are going to be really rich in terms of the abundance of the different microbes in there. But we are really interested in looking at the planktonic microbes in the research that we do. So we do, in my lab, we also do study the sediments, but not in this environment. We were interested in the planktonic bacteria and archaea in the water column.
Looks like we have a question from Jenny.
Yes, hello. Thank you for the nice talk. So I have a question regarding the abundance that you showed of SAPO5, how it changed from oxic to anoxic conditions. You once showed this in form of box plots. And I had the feeling that what was also shown there is that the variability of the abundance changed. I think it was greater under anoxic conditions, if I'm right, and lower under oxic conditions. And I was just wondering if this could be an indicator for other drivers, like, for example, under anoxic conditions that drive the abundance of SAPO5 in your samples, or drove the abundance in your samples.
So I think you're thinking of it may be what it's, for example, if it's a sulfur oxidizer. So I think you're right that what we analyzed is not everything, right? So there are other drivers that could be certainly really important in structuring microbial communities, but in particular driving the abundance of SUPO5. So I think that's probably what you're asking, and we didn't measure. For example, hydrogen sulfide concentration. So that's a really good question, and that's not something we measured. So certainly, we do have to be cautious when we say these are the drivers, because we didn't measure everything, and there are certainly other drivers that are really important when you are talking about microbes, but in particular SUPO5. Yeah.
Okay. And if I may add one more question is. Because like in our project, we also are interested in how mixing of waters contribute to the occurrence of the different organisms. And I was wondering if maybe oxygen, because we have in this area, right, the mixing of low oxic with like more oxic waters. And I was wondering if maybe also the oxygen as a driver, what you saw in your samples, if this could also be a side effect of, for example, mixing of different water.
I love that question, because we actually really thought about this a lot with Lauren's work, because we were if you think about that freshwater, you always hear about freshwater input. It inputs nutrients into the marine environment, right? So you think of nutrients, and then you can see this that it has a huge impact on the marine environment, right? So you have phytoplankton bloom that decays, oxygen decreases. And so, but what else is being introduced from that freshwater input? Microbes, right? So it's entirely possible that other water masses are introducing, you could think of them as sort of a seed bank, right? Where they're introducing new microbes into that water column that weren't necessarily there before. We didn't look at water mass and contribution. That's such a great idea, though, is to see if you are observing different microbes or maybe different abundances of those microbes in the different water masses, and then thinking about the mixing that would occur, and are those different water masses potentially a seed bank introducing new microbes, or maybe microbes with different abundances than what that were there before that water mass was introduced. Yeah, so that's not something we can get at, but that's a great question.
We have one more. I'll take this last question here from Barbara McGregor. It's sort of related. Any idea if species are metabolically active at all depths where they're found, or just surviving? Not easy to tell, I know.
Yeah, that's a great question. So we only did for this project, because we don't have funding for this. We didn't go further and do omics analyses. So that's what I do anyways. There are other ways to get at cell activity, to see if maybe these cells are just, and it kind of actually goes back to what Jake asked, right? Maybe these cells are there, but they're just at such low abundances. And if they're there and they're at really low abundances, are they active? And so we didn't do any, for example, metatranscriptomic sequencing, where we could both attribute the activity to certain microbes, but actually determine if maybe these ammonia oxidizing archaea are active in the NBUS. We do have a lot of evidence that there is active ammonia oxidation in the NBUS from a couple other studies. But they didn't directly attribute that to the AOA that we saw were abundant, but then decreased in abundance when you had higher oxygen concentrations. So we didn't look at activity, but it's absolutely something that you would want to do, and determine whether these microbes that you saw are actually actively carrying out the metabolism that you're attributing to that particular microbe. And we did not do that here.
Lauren, anything to add to any?
No, I think Olivia's doing, that's exactly right. So, yeah.
Cool. Awesome.
But we did in Lawrence, though, we certainly did carry out metatranscriptomic sequencing, and absolutely those ammonia oxidizing archaea are really actively carrying out ammonia oxidation. So we don't have that data for the NBUS, but we do have it for her PhD thesis work.
Great. Kurt, did you want to kind of?
If I may, I would like to ask a question to Lauren. Actually, for both. We have two microbes that solve one problem. One is detoxifying the sulfide in the water column. The other one is converting toxic levels of ammonia, but both just shifted into another problem, which is the N2O production. So what did we win by this?
Oh, sorry, what did we learn?
No, I mean, the microbes are supposed to solve problems.
Oh.
Not just to switch problems from the water into the atmosphere.
Yeah, so they're due to this, producing of N2O, they're just further contributing to potentially warming climate and expanding of oxygen minimum zones. So it's kind of feedback, positive feedback, I guess, could be.
Right,
So
and we
solving our problems.
No, the microbes don't care. They're just going to carry out their metabolism. But they also, you forgot that they are also going to lead to accumulation of nitrite in the water column too, right? So that's another issue. But.
Maybe another one to Lauren. Did your knowledge about the Gulf of Mexico help after the oil spill? I could imagine that eutrophication led to more biomass, organic material that collected the oil, and then it sank down.
Oh, that's so interesting. So I didn't look at the oil spill data, but Olivia Mason was one of the first people out to sample the Gulf of the big oil spill that happened, the Deepwater Horizon. But my samples were collected after that big oil spill, and they weren't anywhere. They weren't really near it. That was in deeper water, and my samples are pretty close to shore. But I know Dr. Mason has more experience with looking at the oil seeps in the Gulf and the influence of the oil spill on the microbial community. She has a few papers out on that research.
Yeah, I mean, we were really worried that the oil spill was going to interact with that low oxygen environment, which would be even more problematic environmentally. But it didn't actually interact with the low oxygen bottom water there.
Thanks very much for the great talks.